These are the lecture notes for Chapter 11 on meiosis and sexual reproduction. So first, a little bit of information on sexual reproduction. Sexual reproduction combines cells called gametes. Most of the time, it would be an egg and a sperm, but not always. But the cells are called gametes. Each gamete comes from a different parent. So the gametes come from two parents, and that combination is called fertilization. The combination of the two gametes. And by mixing chromosomes from two individuals, you increase genetic diversity. So that is the purpose of sexual reproduction. And the difference in sexual reproduction, or the process of meiosis, is different from mitosis, in which one cell produces two genetically identical daughter cells. The difference is that one cell produces four not genetically identical daughter cells. Therefore, the parent cell is diploid and the four daughter cells are haploid. They only contain half of the chromosomes of the parent. So, sexual reproduction involves the process of meiosis. Meiosis is going to be very similar to mitosis, so we'll focus more on the differences, the stages as far as lining up in metaphase and pulling apart in anaphase. Those many of the components or many of the processes that happen in the different stages are the same, but we'll focus on the differences. So most plants and animals have two sets of chromosomes. One set comes from each parent, and that means that they are diploid. Diploid means that they have one set of chromosomes from each parent. In humans, there are 23 sets and 23 pairs of chromosomes. So there are 23 sets of chromosomes from the father and 23 from the mother. So reduction of the chromosomes in meiosis, reducing the chromosome number by half, is necessary because otherwise in every generation you would double the number of chromosomes. So you must halve the number of chromosomes during meiosis so that when the gametes unite and fertilization occurs, the original zygote that's going to develop into the new organism has only two copies of the chromosomes, not 4 or 8, or it would just continue doubling, which would, of course, never work. Eventually, cells would not be able to hold the number of chromosomes because they would double every time. So every time that two individuals mated. So reproductive structures that produce gametes or spores, because in plants they would sometimes, plants and as well as fungi will produce spores that kind of are the same thing as gametes. They're going to have cells with only a single set of chromosomes, so we call them haploid cells. So what's happening in meiosis is that sperm and egg, or gametes or spores, but of course we're just going to focus more on humans, so we're going to assume that it's sperm and egg that are being produced. In meiosis, instead of producing two diploid daughter cells, there are going to be 4 haploid daughter cells. So meiosis reduces the number of chromosomes to half to produce haploid cells. And meiosis is responsible for producing the egg in humans and the sperm in humans, as well as the egg and sperm in plants, and gametes in other organisms, and spores in some organisms. Now, another difference in meiosis and mitosis is that there are 2 cell divisions in meiosis. There's a meiosis 1 and a meiosis 2. So this is a list of the stages that occur during meiosis 1, the first cell division. First of all, you're going to have an interphase at the beginning of meiosis that is exactly the same as interphase in mitosis. You're going to have a G1 and S and a G2 phase. And the main thing that happens during interphase of mitosis, one of the most important things is the S phase. The DNA replicates. All of the chromosomes produce sister chromatids. So now we get to a difference. In prophase 1 of meiosis 1, of that first division, the chromosome pairs, we know there are 23 pairs of chromosomes. 23 of them come from the father and 23 from the mother. Or you could say 23 came from the sperm and 23 from the egg, but of the parents. But I think we can agree there are 23 pairs of chromosomes. And everything that happened in prophase of mitosis happens in prophase of meiosis. But there is an additional thing that happens. This additional step, or this additional component of prophase 1, is that the homologous pairs match up and form tetrads. Sorry about that. I got a phone call. So the main thing, again, that happens in prophase 1 that's different from prophase of mitosis is that the homologous pairs match up. This process is called synapsis. I'm trying to find a place to write this, synapsis. When the homologous pairs match up, it's called synapsis, and what they form is called a tetrad. And then you're going to have prometaphase, which is very similar to prometaphase in mitosis, except this time when the chromosomes start moving toward the metaphase plate, the spindle microtubules are going to attach to the kinetochores. But when the chromosomes move toward the metaphase plate, they're going to move in pairs. So in metaphase 1, you can see that they are lined up instead of in single file like they were in mitosis. They are lined up on the metaphase plate in homologous pairs. The pairs are actually lining up. If this were in a human cell, in mitosis, when you have metaphase of mitosis, you would have 46 chromosomes lined up in a row. When you have metaphase 1 of meiosis 1, you have 23 pairs lined up in a row. Then in anaphase 1, there's another difference. In mitosis, during anaphase, we said that the sister chromatids pulled apart. Sister chromatids do not pull apart in anaphase 1. It is the homologous pairs that separate. The sister chromatids actually stay together. And then in telophase 1 and cytokinesis is very similar. The only thing is that sometimes cytokinesis does not go to completion, and there is definitely not another interphase in between meiosis 1 and meiosis 2. So let's look at meiosis 1 in the notes. Meiosis is preceded by normal interphase. Like I said, just the same as if this were mitosis. Interphase proceeds as normal. And in the S phase, you still have duplication of chromosomes so that now you have sister chromatids, and the sister chromatids are attached at the centromere. However, in prophase 1 of meiosis 1, here's where the difference comes in. You still have the nuclear envelope disappears and the spindle fibers begin to emerge from the centrosomes. You still have the same things happen, except and the chromatin condenses and forms chromosomes. But what's going to happen in prophase 1 is a process called synapsis, where the homologous chromosome pairs, each of the 23 pairs, find each other and match up, and that is called synapsis. And then, because they are so close together, pieces of the chromosomes can actually exchange. For example, the chromosome that came from the father, we're just going to assume that's the blue one, and the chromosome that came from the mother is the red one. Pieces of these, like the ends especially of these chromosomes, they are so close together that they will cross over and they will actually exchange pieces. And this process is called crossing over. So we don't just get genetic variety or genetic diversity from the process of forming gametes, um, the fact that half the chromosomes come from the mom and half from the dad, and that's kind of a random, it's random which chromosomes are come from the mom and which come from the dad. So you get a random kind of assortment of traits from the mom and the dad. We also have more diversity because of this phenomenon of crossing over. And this is a picture so you can understand it a little bit better. Even though crossing over in reality, there's usually more crossing over than what you see here, this is just to help you understand it. So the A's and the B's and the C's represent genes. So if you look in the blue chromosome, it's got a capital A, capital B, capital C, which is going to represent the gene for some trait. And then in the red chromosome, those genes are lowercase. So what that means is the red chromosome has a different version of that gene. So just to give you an example that you maybe could relate to, let's see, in humans, we have, let me think of one that's simple. Okay, well, you can have freckles or not freckles, and having freckles would actually be an F, but we're going to pretend it's A. So having freckles would be a dominant trait, and it would be represented by a capital letter. So we're going to say that capital A is the gene for freckles. Means you're going to have some freckles. Doesn't say how many, but it means you will have some freckles. Little a means that you will have no freckles. So, and we say it's recessive because if you are going to have no freckles, you have to get 2 copies of the little a gene. Now, we will talk more about genes and heredity in further chapters, but this is kind of an introduction. So what you're seeing is in the process of synapsis and crossing over, you can see that the C gene is the one that's going to change places. So what you will get is you will get when eventually when all of the sister chromatids are separated, because they will separate in meiosis too, you will end up getting these possibilities in the offspring, or in the gametes. The gamete, which could be egg or sperm, could either have genes big A, big B, big C, or they could have the genes little a, little b, little c. Or because of crossing over, they could have a big A, a big B, and a little c, or a little a, little b, and a big C. So instead of just 2 possibilities, which is what you would have gotten if there were no crossing over, you're going to have 4 different possibilities, which increases the genetic variety in the offspring. All right, moving on to prometaphase 1. This is very similar to prometaphase in mitosis, except this time the chromosomes, as they are moving to the metaphase plate, they are moving in pairs. So the homologous pairs or homologs are still held together at the chiasmata. And so the tetrads are going to line up at the metaphase plate instead of just in single file. In metaphase 1, tetrads will line up at the metaphase plate. So the homologous chromosomes, or tetrads, will line up at the metaphase plate.